name is Mauricio Bonifacino. I am from Montevideo, Uruguay, and I am part of a team that put together this workshop on compositae organized by the International Compositae Alliance. Hope you guys are doing okay, wherever you are in the world watching, and I thank you for having attended this workshop. So, without further ado, let's begin with this introduction to the family Compositae. Well, okay, in this short introduction to the Compositae family, first I'm going to provide a brief sketch of the phylogenetic context of the family. Then I'm going to explain briefly the main morphological features defining the Compositae. And finally, I'm going to dedicate most of this introduction to explain the very basic of the morphological traits used for both classification and identification in the family. The Asteraceae or Compositae are located in the order Asteraceae in the clade Asterid, and that is an order that is largely dominated by the Compositae, which is by far the largest plant family of the angiosperms. So you see here representation of the sizes of the different families of the Asteraceae, and you could see how conspicuous the Compositae are. I painted in red also the sister family, the Calyceraceae, which is restricted to southern South America, and I'm going to comment a little bit more about that family in just a minute. One of the features that act as a synapomorphy for the whole Asteraceae order is what is called secondary pollen presentation. That is a defining feature of the Compositae itself, but it's also a defining feature of the Asteraceae. So what do we mean by secondary pollen presentation? That the pollen, in order to be accessible for the pollinator, needs two processes to intervene. So in Act 1, the pollen is released inside the anther tube. I'm using here the example of the Compositae, but similar things happen in most of the Asteraceae order. So in the first act, the pollen is released inside the anther tube, and then in the second act, the pollen is pushed outside by the style and made then available to the pollinator, as you see there in the image. And there you can see that pollen that arrives to the flower, it's a pollen that comes from other flowers. So that's a mechanism to prevent self-pollination, given the place where the stigmatic area is located in these plants, which is in the inner sides of the style branches. So this trait is a feature from the whole Asteraceae, but definitely is one of the defining features of the Compositae itself. So the sister family, the Calyceraceae, is a very peculiar group. It has inflorescence that resemble the capitulum of the Compositae, although they are now named cephaloid. I'm going to explain in just a minute what is the difference with the capitulum. They have the filaments of the anthers fused, and they have the anthers free. That is more or less the opposite that occur in the Compositae, which have the filaments free between each other and the anthers are fused. The ovule is apical here, and they are restricted to southern South America. So if you look at the structure of the inflorescence in the Calyceraceae, you will see they are marked in red a terminal floret, and then in green other parts of the inflorescence that are actually part of cymes. This is from a very nice paper by Posner et al. That they call this cephaloid. And if you compare that to the Compositae, you will notice that in the Compositae we don't have that terminal central floret that occurs in the Calyceraceae, and that is a major difference between the two. Even when they have the structure that resembles a capitulum, the way it develops is very different. Okay, so moving on, if we look at the current classification of the Compositae, you will see that it's composed of almost 50 tribes and a substantial number of subfamilies. among which the major tribes are the Asteraceae, the Seneциoni, the Sicori, the Cardui, the Antemidi, Nephali, Vernoni, Helianthi, and Eupatori. All those are the major tribes. So, moving now to the main traits defining the compositae, exemplified here with the sunflower. Well, as we just mentioned, the inflorescence is a capitulum. That is, and this is what is called a pseudantium. That is an inflorescence that resembles a flower. The filaments are free, and the anthers are fused in the florets. The ovule is basal here. It's one ovule per ovary and attached to the base. The calyx is modified into a structure named a pappus that has a pivotal role in the dispersal of the fruits and also in the defense of the fruits. And as regard the distribution, they are pretty much everywhere, although they prefer open habitats and, xeric habitats as well. We don't have much time, but whenever we speak about or try to briefly characterize the family, it is important to mention this French researcher, Henri Gabriel Cassini. He is the father of the synanthropology, that is the study of compositae. Early in the 19th century, through a series of many papers, he methodically undertake the task of studying each of the floral organs at a time across the whole family that was known to him. And in that way, applying that sort of comparative method, he nailed down which were the characters that marked the natural groups. And he is the one who actually defined the original tribes of the family, based for the most part on the styles characteristics and also on stamen and corolla characteristics, but mostly relying on style characteristics. It was a really remarkable person. He did that as a hobby in his after hours time because he was a judge. And you can read, more about him in the Funk et al. Volume on compositae. There's a whole history chapter dedicated to this, and there's a lot of information in Henri Gabriel Cassini. Okay, well, moving on now to see some of the most iconic features of the compositae. Definitely the structure of the head or the capitulum is one of the most striking ones. Here we see a typical yellow capitulum on top view on the left and on inferior view on the right. And these, group of bracts are what constitute the part that is called involucre. Then we look on the left side image. We will see, what it's usually called the marginal florets, and then the disc florets in the center. So a typical capitulum has two types of florets. Ones that are arranged in the periphery and are called marginal florets, and others different than those arranged in the center, and those are called disc floret. As you can imagine, in a family of over 30,000 species, there's a lot of diversity. So this is just the very basic of the structures. As the different presenters cover the different tribes, you will see a whole wealth of diversity in terms of capitula shapes and forms. If we look a little bit further into the structure of the capitulum, we will see this is a longitudinal section of a capitulum, of a typical radiate capitulum. There you see the marginal florets. There you have the disc florets in the center. You have all these florets are in most cases sessile, and they are attached to expanded receptacle that is surrounded by an involucre made of bracts that receive the special term of phylaries in the compositae. In some groups, each floret may be associated with an individual bract that is called a pallia. If we take a closer look at one of the disc florets,
Well, we will see that it's composed by a tubulose corolla, that it has a sort of tubular shape, as its own name will imply. Then we have the style branches visible there. Then we have the Androecium that has the peculiarities that we commented before of having the anthers fused and the filaments free. We could see the ovary. The Compositae have an inferior ovary, so you see all the other parts of the flower attached on top of the ovary. And if we look at the lateral floret or marginal floret, first we notice a corolla that is zygomorphic, contrasting with the actinomorphic tubulous corolla. And then we see this structure that represents a modified calyx that is termed the Pappus. Another interesting feature in the Compositae is the pollen, and the pollen is varied in the family, although the most common type of pollen in the Compositae is the echinolate, but other types of pollen also present in the Compositae are the xylate and the lophate. If we look at the phylogeny by Mandel et al. From 2019, we could see on the tree some interesting morphological patterns with a few major characters. For instance, if we look at the length of the corolla lobes, that is the part of the petals that are free at the apex of the corolla, we will notice that an important group of the family that is called the Asteraceae subfamily has those lobes short. And most of the rest of the family we will see the lobes of the corolla being long. If we look at the stigmatic surface, we'll notice that a good part of the family has the stigmatic surface in two marginal bands, while the rest has the stigmatic surface in a continuous surface. And then if we look at the pollen, the whole Asteraceae has echinolate pollen, and the rest has pollen lophate or xylate. Okay, in those Capitula that we saw, there's a huge variation in the shape of the corollas. We either have actinomorphic corollas like the two ones that we have in the left, that's called tubulose or filiform, and then we have zygomorphic corollas. Starting with the most common ones, that is the true ray. That's, as you can see, has an expanded limb with three apical lobes. Then we have the four lobe that is restricted to some South African Compositae. Then we have the ligulate one, which has 5 teeth in the apex. Then the bilabiate that has 2 lips, an outer lip and an inner lip. And then the pseudobilabiate, in which we also have 2 lips, but the inner lips, instead of having 2 petals, have 1 petal. And the other 4 petals are in the outer lip. So the way the flowers are arranged in the head varies in the family. So for instance, this sort of head or capitulum, which is one of the most commons, is called a radiate capitulum. And in this we see 2 types of florets with 2 types of corollas. We have the marginal florets with true ray corollas and the disc floret with tubular corollas. In another type of capitulum, we could only have what is disc florets, and this particular, so only one type of floret. This type of capitulum is called discoid capitulum. So that type of floret must have actinomorphic corollas in order to be called a discoid capitulum. So there you have the disc florets. Then we have another type of capitulum which is called a deciform. In the deciform we have 2 types of florets with different corollas, but the marginal florets are sometimes hard to be differentiated with the naked eye. It kind of looks like we have 2 different things, but sometimes we are not sure. We need to look close with the hand lens. So there you have the marginal florets, and you can see there this floret. You can see that the apex of the lobes are much larger in the disc floret in this case than in the marginal florets. Some other heads are what researchers call radiant capitulum, in which all the florets have the same type of basic tubulose corollas, but the one that appear towards the periphery of the capitulum have more expanded a part of the corolla. So that it tends to look like a radiate head, but not being a radiate head. this is just a tiny representation of the whole diversity of Capitula that we could find in the Compositae. Another interesting feature regarding the structure of the florets is what part of the floret is fertile. If it's only the Androecium, or only the Gynoecium, or both. So here we could have monoclinous florets like the two ones on the right. One case in which both the Androecium and the Gynoecium is fertile, or the other case, in which only the Androecium is fertile and the Gynoecium is sterile. Then, when we have declinous florets, these declinal florets are only pistillate, and they could be either fertile or sterile. If we take a little step further into the type of Capitula, when Capitula have only one type of floret, they are called homogamous Capitulas. And they could be made of just actinomorphic corolla, like in this case, in these two cases, one in which only have tubulous corollas and the other in which we only have filiform corollas, or a case of homogamous Capitula composed exclusively from floret with zygomorphic corollas, like in this case with only ligulate corollas, or pseudo-bilabiate corollas, or only bilabiate corollas. The other type of Capitula are called the heterogamous Capitulas, of which the radiate Capitula that we already saw is one of the most typical examples. And these heterogamous Capitula could be made of just actinomorphic corollas, like the one on the left, which will correspond what is called a deciform Capitula, with tubulose, florets with tubulose and filiform corollas. Heterogamous with zygomorphic and actinomorphic corollas, like this case tubulose and true-ray corollas present in the same head, or tubulose and four-lobed corollas present in the same head, or like in this case, all of them are zygomorphic corollas. In this case, bilabiate corollas of different shapes. If we pass now to the Androecium, the Androecium has a lot of characters, and some of those were used by Cassini himself when defining the tribe. So let's see what are the main things that we need to look in the Androecium. So we have the filament, and we have the anther. That are the two basic parts of a stamen, right? In the filament, we could have a differentiated part that is called the filament color that could change in the overall shape or in the thickness of the wall of the constituting cells. Another important part of the anther is the projection of the connective tissue that is called the sterile appendage. This could be either present or absent, and it could vary in the size and the shape and the color, and it's an important character at the tribal level sometimes. Another important part that we need to pay attention is the insertion point, where the anther is attached to the filament. Because if you see that yellow part represents the fertile part of the anther. So based on how far extended along the TICA the fertile part is, we have two basic type of anthers. What we call calcarate is when the fertile part extends beyond the insertion point. It extends beyond. In that tail that anther is having, the one on the left, there's pollen there. So this is a typical calcarate anther. When the fertile part doesn't extend below the insertion point, we have what is called an acalcarate anther. We could have tails in those anthers that extends below the insertion point, but notice that in that case that it's just an sterile tail. Those are the main features that you're gonna be wanting to look into the Androecium. I don't have it illustrated here, but there are certain groups in which the color of the anther might provide, identification value. Like for instance, if you see anthers that are all blackish, that means that you are in the Helianthi alliance, where a good amount of groups in that collective of tribes have those anthers. But by far the main character in tribal classification is the style. And with these examples that I have here, I am not even touching the surface of the whole diversity that occur in the family. But the purpose of this slide is just to point out which are the main characters that you will be looking into the styles in order to see and be able to identify the different types. So let's start with the very basic. You will notice that in the Compositae, the styles are branched, showing two branches. That's kind of hinting you to the number of carpels that you have in the ovary. So the point in which the styles bifurcate, that is called the bifurcation point. And that marks two parts of the style. The basal part that is called the shaft, and the upper part that is called the style branch. In some of the styles, you will see that the stigma surface covers the whole inner or adaxial part of the branch. It's continuous, it covers the whole surface. While in other styles, you will see that that stigmatic area is restricted to two marginal bands. This is a major trait that is well correlated in the phylogeny, as we just saw a few slides ago. Another important trait are the pollen collecting trichomes. Those pollen collecting trichomes could be distributed along the style branch and extend below the bifurcation point, like in this case, or they could be restricted only to the style branch and do not extend below the bifurcation point. That is another major trait. Another important trait is the presence or absence of an sterile appendage. If you look in the style on the right, you will notice that the marginal bands of the stigma do not run across the whole style branch. They are restricted to the base. So that denotes an apical part that is sterile. That is called the sterile appendage. The presence of a sterile appendage, the size, the ornamentation, all those provide systematic value in the family. And then the length of the branches is another important trait. With a combination of all these features, you will be able to see many different styles that in most cases define and characterize each of the different groups in the Compositae. So here we have an example of the genus Stevia in the Eupaterae, and there you can see the two marginal bands. Usually the shape of the cells of the stigma is different than in the rest of the style, so you should be able to tell it, even when there's no difference in color, like in this case. And here you can see the sterile appendage very easily as well. Passing on to the fruit that is called a cypsela, and by some an achene derived from an inferior ovary. The main characters that we are going to see in an achene, or a cypsela, are the size of the achene, the shape of the achene, and the type of indiment and distribution. Some of the cypsela could have a beak or rostrum that is a elongated and thin part. We place it at the apex of the fruit. We could also identify ribs that usually represent the vasculature of the ovary. The type of the indiment, the type of particular trichomes that occur, and the distribution, those are important characters. As it is a differentiated part at the base of the fruit that is called a carpopodium, or the foot of the fruit. And then there's another interesting feature that is the deposition of a substance, of a dark substance called phytomelanin that could either be present or absent, and that marks important groups in the family. A trait that sometimes can be hard to see is the number of ribs, especially in those cases where we have a lot of indumentum, covering the fruit. So in those cases, making a transversal section could help you see the ribs, like in this case, for instance. Here we see an example in the Eupaterae where we could easily see the ribs, and you see the phytomelanin that is covering the whole surface of the fruit. And to end this very short presentation on the main features, let me say something briefly about the Pappus. The Pappus is that modified calyx in the Compositae, and the main characters that we are going to be considering when studying the Pappus, well, the most obvious one is the length of the Pappus. Then there's the number of series of the Pappus. In this particular case, we have 3 series. The easiest way to spot this is to make a longitudinal section of the fruit and see how many rows of elements in the pappus we have. In this case, we have just 3. The other main character is the diversity of pappus elements present in that particular structure. So we have situations in we only have one type of element that is called the homomorphic pappus, or we could have 2 types of elements, and we call this sort of pappus heteromorphic pappus. Please notice that the type of element could vary inside the floret, and it also can vary in the same head between the disc florets and the marginal florets. If we look at the particular shape of the pappus elements, we could see that there's a huge diversity of shapes. We have what is called the bristles, which are, very, very thin elements that could be either scabrous, barbellate, or plumose, depending on how long those projections are. Then we can have what is called the capillary bristles, which are bristles but very thin. If you look at the bottom of each drawing, you will see that there's a cross section that illustrates how many cells usually make up that structure. Some of the elements of the pappus could be very, very rigid and strong, and some of them are called awns. And in some other cases, we have elements that are flattened, and they are called palliaceous elements. And they vary on the shape of the margin. They could be either ciliate, fimbriate, or they could have the entire margin, or they could be setose with a prolonged apex. And then another type of pappus are those called coroniform, which are represented by a sort of crown-like structure. Well, that was just a very, very rapid and brief introduction to the main traits of the Compositae. You can find, I want to thank you for your attention, and I want to point out that you could find a lot of information at compositae.org, where we soon will be, uploading the whole Fankeral volume from 2009. Well, there's a wealth of information on all things Compositae. Okay, thank you very much, and I will gladly answer your questions.